So what are carbohydrates? Well, they are a major source of energy that we eat, right? So we consume three types of biomolecules. There's the carbohydrates, the fatty acids, which we'll talk a lot about later in the semester, and then also we eat proteins. We'll talk about a lot about those later on in the semester. But carbohydrates are important because they are the fastest metabolizing energy source that we consume, and they're readily available. Um, they're made of carbon, hydrogen, and oxygen, and they're also known as saccharides, which actually means sugars. Um, so, how are carbohydrates made, like glucose, for example? Well, they're actually made from plants. Plants will take carbon dioxide, water, and energy from the sun, and then using photosynthesis, produce glucose and use that as their sole energy source to make ATP. So when you think about it, right, uh, animals like us will breathe out CO2 during respiration, and then the plants take that CO2 along with water and sunlight and make it glucose. And then when they actually make glucose, one of the byproducts is oxygen. That's why everybody should have a plant in their office or something to increase the oxygen supply in the room. It makes you more energetic. But so this is a process known as cellular respiration, and how we live symbiotically with plants. Because if they didn't make that oxygen when they were making their own glucose, we would not be able to have enough oxygen to breathe. Uh, Actually, that's not true. I think that, well, it is true because it's not really the plants that are on ground, on land, that make most of the oxygen that we breathe. Um, the vast majority of the oxygen, I think it's like 70% of the oxygen that we breathe, that in the air comes from the oceans. And that is because there's number one dissolved oxygen in the ocean, but then also there's a gazillion microorganisms like phytoplankton and dinoflagellates and all those guys. They all do photosynthesis too. So that's where a lot of the oxygen that we breathe comes from. So I guess it's partly true what I said. Uh, that it's just not plants, but it's photosynthesis that provides us with oxygen in the atmosphere to breathe. So, pretty interesting stuff. Next slide. There are three types of carbohydrates. They're the monosaccharides, which are the simplest carbohydrates. They only have one ring. They're the disaccharides, which have two rings, right? And then there are the polysaccharides that have multiple rings, right? Um, Once you get down to the monosaccharide, you can't break down the sugar into any more monosaccharides. But as you can see, going upwards here, a polysaccharide can be broken down into many sugars, and a disaccharide is broken down into a single carbohydrate or a monosaccharide, right? And this happens through a process known as hydrolysis. It uses an enzyme in an acidic solution, right? So polysaccharides can be broken down into monosaccharides, and then disaccharides can be broken down into monosaccharides, both by hydrolysis using an enzyme and acidic solutions. But a monosaccharide is the lowest you can get before you break the molecule so much that it's no longer considered a sugar or a saccharide. So let's talk about monosaccharides a little bit here. Um, so monosaccharides are usually 3 to 7 carbons in the chain, right? And the important thing is that they all contain a carbonyl group, okay? Some kind of carbonyl group. Carbonyl functional group. We learned that a couple of a couple of lectures ago. So if the carbonyl group in the monosaccharide is an aldehyde, then we call the an aldose. That's an aldose sugar, right? If it's a ketone group, then it's a ketose or a ketose sugar, right? So recall what those mean, what each one of those is, right? An aldehyde looks like this. It's a carbonyl with a hydrogen on one side, right? And then a rest of the molecule on the other side, okay? That's an aldehyde. So that's what this is up here, right? And this R group that I just put here, that rest of the molecule, is this part. the rest of this sugar right here. Okay, so that's an aldose. That sugar is erythros, and it is considered an aldose. Okay, remember what a ketone is. A ketone is a carbonyl group that has R groups, or the rest of the molecules, on both sides. So our example over here is urethylose, urethylose. Wow, that's hard to say. Urethylose, right there. It's a ketose. And you can see that one part of the R group here, right? We'll call that R1, is over here. Okay, here's R1, and then the other one, R2, right here, is the rest of this guy right here. So I hope this actually shows you the difference between what an aldehyde is and a ketone, right? An aldehyde has an H on one side, right? A proton or a hydrogen on one side, and then it has the rest of the molecule on the other side of the carbonyl, right? Whereas a ketone has rest of the molecules on both sides. There's no H there, right? I mean, there'll be H's there connected to other things, but it's more than just an H, okay? They all have hydroxy groups on all the carbons except for the carbonyl carbon, right? And it makes sense because the carbonyl carbon doesn't have room for any other hydroxy groups, any other groups, right? So as you look at things like glyceraldehyde, Threose, ribose, and fructose, right? On every carbon that's not the carbonyl carbon, there's an OH connected to it, right? There's 3 carbons right there. That's not the carbonyl carbon. There's another one right there, right? Well, they all have OHs on them, right? OH there, OH there, OH there, OH there. Make sense? If there are 3 atoms of carbon present, then it's called a triose. If there are 4 atoms of carbon present, then it's a tetrose, like tetracycline, or tetra is Greek for 4, just if you didn't know. Pentoses have 5 carbons, and hexoses have 6 carbons. We talk mostly about pentoses and hexoses in biochemistry. That's what we do the most, right? An aldopentose is a 5-carbon monosaccharide with an aldehyde group, and a ketohexose is a 6-carbon monosaccharide with a ketone group, right? So keep all that stuff in mind when we start identifying some sugars throughout this lecture. So when we talk about monosaccharides, we have to discuss structural and stereoisomers. So what is a structural isomer? A structural isomer has the same molecular formula but has a different bonding arrangement. Okay? So here we have the example of C2H6O. That can be arranged into ethanol or dimethyl ether. Same can be said of C3H6O. That can be arranged structurally into propanal and propanone. Those are all considered structural isomers. Well, then what are stereoisomers? Stereoisomers are similar to structural isomers in that they have identical molecular formulas, right? But they aren't structural isomers. Otherwise, we'd call them that. They are not structural isomers. But the important thing about them is that the atoms are bonded in the same sequence, right? But they differ in the way they're arranged in space, right? Where they're arranged in space, and we'll take a look. We'll take a more closely look at that in a second. So when we talk about stereoisomers, there's a couple different types, right? Or a couple ways of describing them, and we describe them such as in terms of their mirror images, right? If their mirror images cannot be completely matched, they are described as non-superimposable, which I'll show you a graphic that describes that in a second, or that shows that in a second. And also, they are known as chiral, right? Chirality is important, especially when we're talking about biochemistry, active sites of enzymes, and things like that. So, chirality is important. So important that it's the next slide. So, what are chiral molecules, right? Chiral molecules have the same number of atoms, but again, they're arranged in different space, right? Differently in space. And they are non-superimposable images, just like we said a second ago, just to reiterate. So a good example of a chiral molecule, or something that is chiral, is your hands. Your hands are identical when you see them, when you put them, they're mirrors to each other like this, right? Because when you put both your hands together, like this, they're mirrors of each other, right? But when you try to superimpose them on top of each other, it doesn't show that that they match anymore, right? So mirror image, they look the same, your hands, right? But then when you try to superimpose them on top of each other, they're not the same anymore. And that's why your hands are chiral, right? A lot of things can be chiral. Gloves can be chiral too because they match your hands. And then your shoes or your feet are chiral. But say you have a mirror image, right, that is identical, but they are superimposable, then they're known as achiral. Okay? They're known as achiral. So an example of that would be these shot glasses right here, which many of us are very familiar with from spring break, right? Shot glasses. I think those are actually doubles. Yeah, those are doubles. Anyway. So we're now talking specifically then about chiral carbon atoms, okay? So these are some criteria for chiral carbon atoms that you need to remember, okay? At least one or more, so a molecule is chiral when at least one or more chiral carbon atoms are present, right? And that carbon atom, to be chiral, has to be bonded to 4 different groups, right? It could be 4 different atoms, or it could be 4 different groups of R groups, right? Like rest of the molecules. They can't be superimposable images, right? Mirror images, kind of like your hands, right? Their mirror images of each other is identical, but they can't superimpose on top of each other. And then the mirror image of the chiral compound cannot be superimposed. I don't know why I put that twice. That's okay. It's important. That's why it's on there twice. So when stereoisomers cannot be superimposed, they're also called enantiomers. Okay, they're also called enantiomers. So here's a really good example here. We have our carbon in the center here, and a carbon in the center here, right? And as we look at it in this mirror that we have right here, it's actually identical mirror images. See, the chlorines and the chlorines are on the same side. The iodines and the both iodines are on the same side. The hydrogens are both pointing outwards towards us, right? And the bromines are both pointing back in the mirror image, just like your hands would be, right? But when you take them and put them on top of each other, notice that the chlorine from the molecule that's on the right is above the bromine, and then the bromine from the molecule on the right is above the chlorine from the molecule on the left. So that's what is that's what it means when they say non-superimposable, right? And so these would be called enantiomers. So let's talk about achiral carbons then. Achiral carbon atoms, they have mirror image of each other, right? Again, that are the same, right? Both of these bromines are on the same side of the carbon there, right? And then the chlorines are on the same side. Both hydrogens are pointing out at you, right? But then when you take out the mirror, right, you get rid of the mirror and you just rotate the one that was on the right around, it actually looks like they're the exact same molecule because both bromines are on the back side over here, right? That were on the back side over here are now over here just like they are for this guy. And then the chlorine is still the same spot, right? And it was in the same spot, and the hydrogen is pointed upwards. So that's what it means to be achiral, where they have identical mirror images, right? But they can be superimposed on each other just by rotating, right? Just by rotating. That's what achiral means. So let's do a little practice here. And let's see here. We have this molecule here, and we're asking ourselves, is this guy right here chiral or achiral? Well, the first thing to note is that it has 4 different groups. So to be, one of the first things you have to understand, not understand, but one of the first things that you have to remember is that to be even chiral, you have to have 4 different groups, right? And so these are 4 different groups. And if you were to see a mirror image of it, right, would it be chiral or achiral? Well, it would be chiral. Over here, look, this molecule has 2 of the same group. That automatically makes it achiral. And then again, 4 different groups. That makes it chiral. Pretty simple. Pretty simple. So that takes us to talking about Fisher projections. So Fisher projections are 2-dimensional representations of molecules of any molecule, but specifically we're using Fisher projections here to represent our sugars, right? Our carbohydrates that we're talking about. Okay, and so basically in a Fisher projection, okay, vertical lines take the place of dashed bonds, right? And if you remember what a dashed bond is, it's when the bond is going backwards, like into the board, right? Or into the screen, because it's going backwards. I know I'm pointing upwards there, but I'm going into the wall, right? And so if you have a dashed bond and you change it to a Fisher projection, it turns into a straight vertical line, okay? And if you have horizontal lines, those represent the wedge bonds. And remember, the wedge bonds means that it's coming out at you, not going back into the board when you're looking at it, or the piece of paper. So those are going to be horizontal lines, right? Horizontal lines. So now that we've discussed the Fisher projections, we now need to turn our attention to D&L notation. D&L notation is talking about D&L isomers. And that designation is based on the chiral carbon that is furthest away from the carbonyl group. Okay? So if we look at this erythrose down here, the carbonyl group is up here. Right? And we want the chiral carbon that is furthest away. So some of you might think that it's this carbon right here, but remember that can't be a chiral carbon, even though it's the first carbon in the line. It can't be a chiral carbon because there's 2 H's on this carbon, right? There's 2 of them. And that alone violates the criteria that it has to be 4 different groups. But if you move to the next carbon, the second carbon, it's the furthest away from the carbonyl group, and it's the chiral carbon because it has 4 different groups. It's got this methoxy group here. It's got this hydrogen here, this OH here, and it's got this entire other part of the molecule here. So that's the very first, the very furthest carbon that's chiral from the carbonyl group. And so when you have the OH on the left side, then we call that the L isomer, right? OH on the left. But if you have the OH on the right side, it's called the D isomer. Okay? And I'll tell you right now, and this is important to know, that in nature, the D isomer of sugars is most prevalent. Nature makes D sugars or D carbohydrates, right? Make sure you know that. So, looking at these two guys, these two images, these two molecules to the right here, this one, same thing. We have the methoxy down here, right? Which means that that can't be the one that's furthest away. That's still chiral. We look at this thing here, the second one. That's the first carbon, the furthest chiral carbon from the carbonyl group. Oh, that's a tongue twister. Furthest chiral carbon from the carbonyl group. There we go. And the OH is on the right side. So this is the D isomer. And then on this one, the OH is on the left side. So that's the L isomer, right? Pretty simple, pretty simple. Okay? So chirality, it turns out to be super important in biochemistry because these enantiomers are either active or inactive in biological systems, right? Why is that? Well, because enzymes and surface receptors on cells, right, which participate in metabolic reactions, right, they're also chiral themselves, right? And the active sites in which these molecules bind in the enzyme, right, are chiral. And what that means then is that they have to fit properly for the enzyme or the surface receptor of the cell to be active. Okay? So if you look over here at this example right here, in example A here, we have chiral receptor sites on an enzyme surface where it's the shapes are going to be your R groups, right? Your different constituents that make them chiral, right? Because we do have chiral carbons up here, pentagon, sphere, cone, and I don't think that's a cone actually. I think that's actually a pyramid. Pyramid and square, right? That's definitely chiral. And the active site on the enzyme surface is a circle, a square, and a triangle. So a molecule that's chiral having the arrangement circle, pyramid, and square is the only one that can fit into these shapes to become active. Because if you look over here, if you have a square on this side and you have a sphere on this side, but then still have the pyramid still in the middle, the only thing that can actually fit in the active site properly is the pyramid. The square and the circle, or the square and the sphere don't. The cube and the sphere don't, making it inactive, right? So one of the most common and well-known chiral molecules used in medicine is, and we actually use a lot in biochemistry too, is this thing called L-DOPA. Right? L-DOPA. And L-DOPA is used to treat Parkinson's disease, right? Interestingly, by the way, L-DOPA is L,3,4-dihydroxyphenylalanine, right? So it's an amino acid, okay? We'll talk a lot about amino acids later. Used to treat Parkinson's disease. Interestingly, only the L version of L-DOPA, the L version of DOPA, right, is the one that is active. The D version of DOPA is not reactive. It's not active, right, in treating Parkinson's disease. Where's this chirality we're talking about here? See, it's right here, right? The star denotes where the chiral carbon is. See? And all these constituents are in the wrong places, right? So you wouldn't be able to fit into the active site to actually make this drug active. Okay? So if you recall, back in when I was talking about sugars, I told you that D sugars, D isomers of sugars, are the most prevalent in nature. Nature makes D isomers of sugars. Well, guess what nature makes when it comes to amino acids? In which isomer it uses? Well, the clue is the fact that L-dopa is the only one that's active, right? In treating Parkinson's disease. It means that nature likes making L versions of amino acids, right? So, as you go through biochemistry and learning about amino acids, every amino acid you talk about that's in nature will be an L amino acid or an L isomer of an amino acid, right? Whereas when we're talking about sugars or carbohydrates in nature, in biochemistry, there will always be D isomers, right? Just remember that. That takes us to Hayworth structures. All right. So the most stable forms of pentose and hexose sugars, which are the 5-carbon and the 6-carbon sugars, is when they become a ring, right? They're not very stable when they're straight chained, right? They're not very stable when they're straight chained. And so what happens is that the straight chain will cyclize, right? And in solution, it's a mixture of the the alpha version of glucose, for example, and then an open chain glucose and the beta version of glucose. So what does alpha and beta mean? We're already just talking about the L and the isomers, right? Well, alpha and beta refer to the hydroxyl that's on this carbon. Right there. Those two. Right? When the hydroxyl is pointed down, it's considered the alpha isomer. And when the hydroxyl is pointed up, it's considered the beta isomer. Right? So this one right here, it's pointed down. So that's the alpha isomer. Right? And both the alpha and the beta versions of D-glucose are prevalent, right? 36% of the alpha, 64% of the beta. So both are very prevalent in nature. The thing that's not prevalent in nature is the open chain, only trace amounts, right? In solution when they're going back and forth like this. And remember, the reason why is because their open chain version is just not very stable. They prefer to be in their closed chain, right? And so, in a Hayworth projection, a Hayworth structure is a straight chain sugar that has been cyclized, right? So how do we draw Hayworth structures? How do we know where things go, right? Well, let's take a gander. So how do we turn a straight open-chained sugar into a Hayworth structure? Well, quite simply, the first thing to do is to rotate that straight-chain sugar, open-chain sugar, 90 degrees clockwise, right? Put it on its side. Make sure that the aldehyde group is on the right side, right? To the right of the molecule, right? And the CH2OH is on the left side. Then, after that, you're going to want to fold that straight chain in half so that the CH2OH group is now close to this carbonyl group, right? Close to that carbonyl group. Then you want to rotate it. This is a single bond right there, right? So the carbon, the chiral carbon right here can rotate its bonds. So you want to rotate it so that the CH2OH group goes up here to where the H is. The H can then rotate to where the OH is, and then the OH rotates up to where the CH2OH used to be. And that's what you have here on this molecule. Why do we have to do that? Well, we have to do that because now it puts this hydroxyl right here in prime position so that the oxygen can form a bond between it and the carbon. Okay? Between it and the carbon. Then this hydrogen left over here, right, goes into forming the hydroxyl that's right there. And then the H was already there before. So if the hydroxyl ends up pointing down, recall, it's an alpha isomer. If it's pointing upwards, the hydroxyl that was made, then it's a beta isomer, right? And just to reiterate, in nature, and when these sugars are in solution, for example glucose here, they're going back and forth between being the alpha version and the beta version, right? With very little trace amounts being the open chain. It can be either one of those when this oxygen does the attack to form that bond between it and the carbonyl carbon. So let's take a closer look at the Haworth structure of fructose. Right? So fructose is a ketohexose, right? But it's different though because it forms a 5-atom ring structure, right? Instead of a 6-atom ring structure like glucose did in the previous slide, right? In the previous slide. And the reason why it does that is because since it's an aldehyde, since it's not an aldehyde or an aldose, right? The carbonyl carbon doesn't have an H over here, right? It actually has an extra carbon, CH2OH over here. That's why it's considered a hexose, because it has 6 carbons. That makes it so that the carbonyl group is actually the second carbon. The carbonyl carbon is actually the second carbon in the chain, right? And so when we do the same thing again, where we fold it in half and then the hydroxyl group attaches itself to the carbonyl carbon, you now come up with a 5-ring sugar instead of a 6-ring sugar. And that's fructose. Okay? And when that happens, you have the OHCH2 on both sides up here. But again, same rules apply. Whenever you have the OH at the bottom that's formed, it's an alpha. When it's at the top, it's a beta. So one of the things that we forget to talk about, or I forgot to talk about in the last slide, is the fact that in Haworth projections, you have this extra bolded bonds right here, right? That they get fat as they go downward here, right? And there's the fattest line, the boldest line, is the bottom one. Well, what that means is that these bonds and these 2 carbons right here are coming out of the paper towards you, right? You've got to think about this in terms of 3D. So let's practice a Haworth projection. Or I always say projection for some reason. Haworth structure. Okay? What's the first thing to do? Turn it on its side. You don't have to, but it's more fun if you turn it on its side. So, like that, right? There's the CH2OH group. And this is a galactose. So it has 1, 2, 3, 4, right? And then the carbonyl, like so. And its arrangement is hydrogen, OH, OH, and then H, and then OH, H, H, OH, right? That's it on its side. Then we want to fold it. The aldehyde is at the end here, right? Which means it's not going to be a 5-member ring. It's going to be a 6-membered ring. So let's fold it. Like so. Here's the carbonyl group, right? In this case, it's the aldehyde. Okay. And then I'm going to go ahead and just put the OH there, rotate it already. Oops, that should be an H. Sorry, that should be an H there. That's wrong. And then OH. H. OH. And then OH. And H. Okay. So we have the electrons on this oxygen right here, and it goes and forms a bond between the carbon and the itself, between the carbon and itself. And then that makes it so that the electrons that were in this double bond go to this oxygen here. And so now this oxygen has extra electrons from the double bond, and those electrons can come over and take that hydrogen. Right? So what are you left with then? You're left with your cyclized galactose, in which I forgot to draw the oxygen, so let's draw that again. Your cyclized galactose. Like that. H, OH, H, OH, H, OH, H, OH, CH2O2, OH, and H, right? But remember, I have this one down here with OH pointing down, so this is the alpha version of D-galactose. But it's really going back and forth between D-galactose and alpha D-galactose and beta D-galactose. So we have to draw that one too. Except I forgot to draw the oxygen again. This was a problem with me when I was in school too. Drawing sugars, I always forget to draw the oxygen inside the ring. There we go. So this one has the OH on top. H. Everything else is the same though. CH2, 2OH. I think I'm writing a little bit too fast for the Apple Pencil again. OH. H. OH. H. H. OH. Right? So that one is the beta version of D-galactose. Okay? It's just as easy as that to turn a Fisher structure into a Haworth structure. So just a quick review, right? It went from the dash wedge structure of the sugar, dashed wedge structure. I don't know why it's not working here. There it goes, wedge structure. We turn that into the Fisher I think I spelled that wrong. There should be a C in there. Let me look. Yes, there is a C there. Right there. Here we go. The Fisher projection. And then from the Fisher projection, we turned it into the Hayworth structure. The Hayworth structure, right? And that's how we do that.